Okay, first things first, make sure you put your name on your paper. Okay. Now we're going to move into pedigrees. So we've worked a bit with Punnett squares, but in reality, scientists rarely use Punnett squares to help them solve genetic problems in human genetics. Punnett squares are very useful for plant and some animal genetics, but not for human genetics. And the reason why is because you can't ethically cross humans in a laboratory and see what offspring they produce. Because think about it. When Gregor Mendel was discovering the laws of genetics, he took pea plants and he intentionally crossed pea plants with one trait with a pea plant with another specific trait. You can't do that with humans. You can't say human A has to have children with human B. So, for the human population, we often use pedigrees instead of Punnett squares. How does this work? Well, scientists often get data from a family that is affected by a possible genetic trait, and they try to figure out how that trait was inherited. Genetic counselors. Now, remember, there was a genetic counselor in the movie that we watched, Gattaca. But it's not a made-up profession only for a movie. Genetic counselors are real. And, for example, if a person knows that they are a carrier for sickle cell anemia, they might go to a genetic counselor to figure out if what the chances are that they would have a child with sickle cell anemia. Or if a family has some possible genetic trait that's affecting them and they don't know how it's being passed from one generation to the next. They don't know if it's X-linked, or they don't know if it's dominant or if it's recessive. Then they might go to a genetic counselor to try to figure out the answers to those questions. So what is a pedigree? Pedigree is this chart that you see over to the right. In a pedigree, a marriage line connects parents in a family. Now, based on when pedigrees were invented, this line is called a marriage line. Okay, you need to fill in your box. It's already filled out on my paper, but you need to fill in your box that identifies this as a marriage line. I realized that genetically, two people do not have to be married to have a child. But like I said, given when pedigrees were developed, that's what this is called. In reality, this line simply connects two individuals that have had a child together. Just be aware, they may not be legally married, but this line is indicating that they have a child together. A child line below the parents shows the siblings in the order of their birth. This is a child line, and it shows the siblings in the order of their birth. Male individuals are represented with squares, and female individuals are represented with circles. A key is used to show what type of shading represents the trait in question. So we're going to talk more about that when we go through this specific example. Generations are labeled with Roman numerals, and individuals are labeled with numbers. Okay, so for example, this person here would be Roman numeral I dash the number 1. Because they're on the Roman numeral I generation, and they are individual 1. This person would be Roman numeral I dash 2. You try it with the bottom row. The only reason why I used the yellow there was just so that you could actually see it, since that middle circle is filled in. So on the bottom row, we have individual II1, individual II2, and individual II3. Okay, so let's figure out how this works. So a little review. An autosomal trait. So we talked about X-linked or sex-linked traits recently. Right now we're going to talk about autosomal inheritance. So an autosomal trait is a trait that is caused by an allele in a gene on an autosome, which is really just any chromosome that is not the X or the Y chromosome. Remember, you have 23 pairs of chromosomes total. You have 22 pairs of autosomes. Then you have the 1 pair of sex chromosomes. Okay, so an autosomal trait example. So we're going to deal with an autosomal trait here. Dry earwax. So there are 2 types of earwax. There's dry earwax and there's wet earwax. Okay? Dry earwax is autosomal recessive. Now remember, we use a lowercase letter to represent a recessive allele. So we're going to use a lowercase e. All right, I'm using E for earwax. Okay, the genotype of an affected person, so this is an autosome, so we only have to deal with E's. We don't have to deal with the X and Y chromosome here. So going back to the simpler notation, it is recessive. So that means we need 2 copies of this allele in order to exhibit the trait. So in order for a person to have dry earwax, they're going to have to be little e, little e. Okay, the autosomal dominant trait would be wet earwax. So wet earwax is a capital E. And the genotypes for a person with wet earwax, well, a person could be big E, and that person would absolutely have wet earwax. But that person, you could also have another person who is big E, little e. Remember, we always put the capital first. And since the big E is dominant, a person who is heterozygous, meaning big E, little e, is going to have the dominant trait. So they're going to have the wet earwax, even though they only have one big E allele. Okay, so now let's use the information that we have to complete the pedigree. So we said that a key is used to show what type of shading represents the trait in question. So in this case, our key states that if the square or the circle is filled in, that means that the person is affected by dry earwax. Okay, and remember, dry earwax is the recessive trait in this case. Okay, so now we're going to go through and we're going to put the genotypes or possible genotypes of the individuals next to each individual. So individual I1 is not affected with dry earwax. Individual I2 is not affected with dry earwax, but one of their children is. So let's start there because that's going to be the easiest place to start. We know that individual II2 is affected with dry earwax, which means she, it's a circle, so it's a she, has to be little e, little e. There's no other genotype that she could be if she's affected by dry earwax. She has to be little e, which means she has to have been given a little e from each of her biological parents. Now, since neither one of them is affected by dry earwax, that means they can't be little e. But they do both have to have at least one little e in order to pass it on. So that means we have just figured out that both biological parents have to be big E, little e. Because it's the only way that they could each pass on a little e and yet still have a big E so that they don't have dry earwax themselves. Now, we know that both parents, we figured out that both parents are heterozygous, which means there's a 50-50 chance that the father will pass on a big E versus a little e. And there's a 50-50 chance that the mother will pass on a big E versus a little e. Because of that, we don't know for sure what the genotype of individual II1 or individual II3 is. We know that they're not little e because they're not affected by dry earwax. But we're not sure whether they are big E, or big E, little e. Could be either one, and we don't know, and there's no way for us to know with just this amount of information. The same goes for individual II1. She could be big E, or she could be big E, little e. Now, if we had more information in this pedigree, we might be able to figure out what the genotype is for those two individuals. Because if they had children, if either one of them had a child that was affected with dry earwax, that means they had to have been able to pass on a little e allele. Now, if none of their children were affected, it doesn't mean that they don't have a little e. So we still wouldn't be able to say for sure what their genotype was.